Hello students, today we are going to study the anatomy of root. Anatomy is defined as the study of internal structure of various parts of an organism. In plant anatomy, basically there are three tissue systems: epidermal tissue, ground tissue and vascular tissue. Now I am going to talk about the anatomy of Dicotroot. Usually Dicotroot example is Bengal gram or sunflower. We have to take the cross section of Bengal gram or sunflower root and we have to study the various parts of this Dicotroot. The outermost layer of Dicotroot is epiblemma, and epiblemma is made up of large cylindrical cells which are compactly arranged, and there is no chlorophyll in the epidermal cells or epiblemma cells. In epiblemma, here and there are some unicellular extensions what are called root hairs, and these root hairs help in absorption of water. Next to this epiblemma there is a cortex. Cortex is made up of many layers of polygonal cells which are of parenchymatous type, and the cells are loosely arranged with a lot of intercellular space. Usually in the roots, starch is stored in the cortical cells. So next to this cortex, the innermost layer, that is endodermis. So endodermis is known as the innermost layer of cortex. Endodermis is made up of cylindrical cells which are compactly arranged, and in the endodermis the radial walls are thickened due to the deposition of suberin. So these suberized radial walls appear like dark bands what are known as Casparian bands, because it was first studied by the scientist Caspari. So the presence of Casparian strip or Casparian band is the striking characteristic feature of Dicotroot. Then the innermost layer or centralmost region of the Dicotroot, that is called as stele. Stele is the region where the vascular tissues are arranged. Outermost layer of stele is called pericycle, and the pericycle is made up of a single layer of compactly arranged simple cells. So in the stela region we can see the arrangement of xylem and phloem. Xylem is the water conducting tissue and phloem is the food conducting tissue. So usually the vascular bundles in Dicotroot is defined as radial, endarch, closed, and it is a tetrarch in condition. So radial vascular bundle means when the xylem and phloem are arranged on different radii alternating with one another, it is called as radial vascular bundle. Generally in the roots there is a radial vascular bundle, and in the stem it is conjoint vascular bundle. So in the xylem strand, the first formed metaxylem is facing the pith and protoxylem is facing the periphery. So this condition is called as exarch condition. So between xylem and phloem there is a non-vascular tissue what is called as conjunctive tissue, and this conjunctive tissue helps in secondary growth. The centralmost region of the stele is called as a pith, and pith is made up of simple parenchymatous cells where the cells are polygonal in shape and they are arranged with a lot of intercellular space. So the vascular bundle in Dicotroot is radial, exarch, closed, and it is tetrarch in condition. Tetrarch means 4 strands of xylem alternating with 4 groups of phloem. So this is about the anatomy of Dicotroot. Coming to the anatomy of monocot root, generally maize or sugar cane is given an example for the study of monocot root. In monocot root, the outermost layer is epiblemma, and some of the epiblemma cells have produced unicellular extensions what are called as root hairs, and these root hairs help in absorption of water. So next to this epiblemma there is a cortical tissue, and cortical tissue or cortex is made up of simple parenchymatous cells. So the cells are arranged with a lot of intercellular space. So the innermost layer of cortex is called endodermis. The endodermis of monocot root is slightly different from that of the endodermis of dicot root. In dicots, only the radial walls are thickened due to the deposition of suberin, whereas in monocots, both the radial walls and the inner tangential wall are thickened due to the deposition of suberin. So, in monocots, we can see U-shaped thickening in the endodermal cells. So, here and there in the endodermis of monocot, we can see there are some thin-walled cells what are called as passage cells. And these passage cells help in the transport of water molecules. Water usually is absorbed by the root hair, and from the root hair then to the epiblema. After passing through the cortical cells, then the water molecules reach the endodermis. And in the endodermis, the water molecules cross the passage cells and finally they reach the xylem elements. After reaching the xylem elements, water molecule is transported through the xylem elements in upward direction. Coming to the Stila region, the centralmost strand or centralmost cylinder in the monocot root is known as a stele, and the outermost layer of a stele is pericycle. So pericycle is made up of single layer of cells which are compactly arranged. There is no specialization or no thickening in the cells of the pericycle. The vascular bundles are defined as polyarch, exarch, closed type of vascular bundle, and of course in the monocot root also vascular bundle is radial. So collectively we can say radial, polyarch, closed and exarch condition in the monocot root. So here there is only single large metaxylem element with the two small protoxylem elements. Just opposite to the protoxylem elements, there is a passage cell in the endodermal layer. So between xylem and phloem, there is a phloem tissue. So between xylem and phloem, there is a conjunctive tissue, and this conjunctive tissue helps in secondary growth. Pith in case of monocot root is very wide, or we can say it is very conspicuous. Whereas in case of dicots, we have seen pith is very small or highly reduced, whereas in monocots pith is very large and very conspicuous. The presence of U-shaped thickening in the endodermis is one of the important characteristic feature of monocot root. This is about the anatomy of dicot and monocot root. Let us study the difference between monocot and dicot root in the next class. Thank you.